I'm Jane Clifford Azizkhan. I'm professor and chair of biochemistry and molecular biology at the Drexel University College of Medicine.
And my name is Oleg Alekseev, and I'm an MD PhD student in Dr. Clifford's lab.
The video that Oleg and I have prepared for you demonstrates the ex vivo culture of corneas obtained from rabbits and human donors. We have utilized these corneas for studying herpes virus infection, and they are certainly applicable for other diseases of the cornea, for study of other diseases of the cornea. And it's a particularly relevant ex vivo model as compared with cells in monolayer culture.
Herpes keratitis is one of the most severe pathologies associated with the herpes simplex virus type 1. It is currently the leading cause of cornea-derived and infection-associated blindness in the developed world. Herpes keratitis is traditionally studied in two types of experimental models. The in vitro model, in which cultured monolayers of corneal epithelial cells are infected in a petri dish, offers simplicity, a high level of replicability, fast experiments, and relatively low cost. On the other hand, the in vivo model of herpes keratitis, in which animals such as rabbits or mice are inoculated directly in the cornea, offers a highly sophisticated physiological system but has higher costs, longer experiments, necessary animal care, and a greater degree of variability. In this video article, we provide a detailed demonstration of a new ex vivo model of acute corneal epithelial HSV-1 infection, which combines the strengths of both the in vitro and the in vivo models. The ex vivo model utilizes intact corneas organotypically maintained in culture and infected with HSV. The use of the ex vivo model allows for highly physiological